Welcome to this explainer. Today we're diving straight into a fascinating, completely invisible world that's operating all around us and, well, literally inside us right now. We're going to uncover the mechanics of bacterial gene sharing. It's basically a hidden microbial information economy, a biological superpower that drives evolution, adaptation, and honestly, one of the absolutely most pressing medical challenges of our time.
So, right off the bat, you gotta wonder, how exactly do bacteria evolve so incredibly fast? You know, when a new environment or threat pops up, they seem to adapt almost instantly. It's like magic. But the answer isn't traditional reproduction, where traits are just slowly, painstakingly passed down from generation to generation. Nope. The secret actually lies in a literal underground information economy where survival codes are traded rapidly, right on the fly.
Okay, let's dive into this and look at our roadmap for demystifying this superpower today. First, what is horizontal transfer? Second, 3 ways to share DNA. Third, the power of plasmids. Fourth, the antibiotic resistance crisis. And finally, biotechnology and the future.
Let's start with Section 1. What is horizontal transfer? Think of it as the microbial peer-to-peer network. We really need to stop framing bacteria as these isolated little loners. Instead, picture them as highly connected nodes in a massive network. Horizontal gene transfer, or HGT, is the formal term for this. It's the movement of genetic material between organisms that are not in a parent-offspring relationship. Unlike vertical gene transfer, passing traits from parent to child, this is happening directly between totally unrelated organisms. Actually, sometimes they aren't even the same species. By skipping the whole reproductive cycle entirely, bacteria use this process as nature's ultimate open-source network to rapidly download brand new traits, like antibiotic resistance, virulence factors, or even crazy new metabolic abilities.
And this brilliantly illustrates exactly why this process is just so formidable. When you stack horizontal up against vertical gene transfer, the contrast is night and day. Vertical transfer is slow, it's limited by generational speed, relies on reproduction, and honestly offers pretty limited genetic diversity. But horizontal transfer, it moves laterally between living organisms at lightning speed, creating massively high genetic diversity. This fundamental difference is exactly why superbugs can adapt to our medicines so terrifyingly fast.
Moving on to Section 2. 3 ways to share DNA. Let's look at the actual mechanics of this microbial trading.
So we know bacteria are sharing this information, but how exactly are they pulling it off? Well, the crucial point is summarized right here with a quick revision of the 3 primary mechanisms they use to swap their genetic secrets. We've got transformation, which is the uptake of free DNA, transduction, which is a virus-mediated gene transfer, and conjugation, the direct cell-to-cell transfer. Let's break down each of these 3 distinct methods so you can see exactly how they operate in the wild.
First up is transformation. You can basically think of this one as a microscopic scavenger hunt. When bacteria die, their cells break apart, and they literally release fragments of their DNA into the surrounding environment. Then, living, so-called competent bacterial cells come strolling along and just absorb these free DNA fragments. They're upgrading themselves using the genetic debris of the dead. If that dead bacterium happened to have an antibiotic resistance gene, the living one can absorb it, integrate it into its own genome, and boom, suddenly it's resistant itself.
The second method is transduction. I like to frame this one as an accidental genetic delivery service. It relies on bacteriophages, which are basically viruses that specifically target and infect bacteria. Sometimes, when these viruses are replicating inside a bacterial host, they make a little mistake. They accidentally package up a piece of the host bacterium's DNA instead of their own viral DNA. So when that virus goes to infect the next bacterial cell, it unwittingly injects the previous bacteria's DNA, sometimes transporting incredibly vital survival traits or virulence genes directly to a completely new host.
And the third method is conjugation. This is the direct handoff. It requires actual physical contact between 2 bacterial cells, usually connected by this tiny bridge-like structure called a pilus. One cell copies a piece of its DNA and sends it right across this physical bridge into the other cell. This specific method of direct cell-to-cell contact is absolutely highly important and is recognized worldwide as a major driver in the spread of antibiotic resistance.
Which brings us to Section 3, the power of plasmids, or as I like to call them, nature's USB flash drives. Now that we know how the transfer happens, what exactly is the cargo? What's being transferred? Plasmids are the primary delivery vehicles making a ton of this trading possible.
By definition, plasmids are circular DNA molecules capable of independent replication. They sit completely separate from the bacteria's main chromosome. Once a plasmid gets transferred into a new bacterial cell, say through that conjugation bridge we just talked about, this foreign DNA can either hang out independently and replicate on its own, or it can plug right into the host's main chromosome for stable, long-term inheritance.
So, what kind of software is loaded onto these microscopic flash drives? Well, plasmids carry incredibly valuable survival traits. We're talking the codes for antibiotic resistance, allowing bacteria to just shrug off our medicines. They carry instructions for toxin production, which makes pathogenic bacteria far more dangerous to us. They offer metabolic advantages, letting bacteria digest completely new nutrients and take over new ecological niches. They even provide resistance to environmental stress. It's a highly, highly valuable package of upgrades in this microbial economy.
Let's look at Section 4, the antibiotic resistance crisis, the real-world stakes. Let's move to and see how this builds into an urgent global crisis, because this invisible swapping of traits has very visible, very real consequences for us. The rapid spread of resistance works in a terrifyingly fast domino effect. Step 1. A single bacterium somewhere develops resistance to an antibiotic. Step 2. That resistance gene gets loaded onto a plasmid. Step 3. Using conjugation, that bacterium transfers the plasmid directly to surrounding entirely different bacteria. Step 4. Those newly resistant strains multiply. It goes from a single resistant cell to an entire resistant population almost instantly.
The ultimate result of this mechanism is pretty stark. Because bacteria are constantly sharing these plasmids back and forth, we are seeing the rapid emergence of multi-drug-resistant bacteria, the superbugs. By swapping these traits laterally, pathogens are increasing in virulence and creating a reality where infection control in hospitals and communities is becoming incredibly, incredibly difficult. It's a direct, real-world consequence of horizontal gene transfer.
That brings us to our final section, Section 5, Biotechnology and the Future, Harnessing the Power.
But hey, it isn't all bad news. Scientists are actually learning how to co-opt this brilliant microbial trick for our own benefit. Horizontal gene transfer has provided some truly incredible tools to human science. Because we understand how bacteria uptake foreign DNA, we use it as the absolute foundation for genetic engineering and recombinant DNA technology. We purposefully use plasmids as cloning vectors to insert beneficial genes into cells for medicine and agriculture. Plus, studying these exact mechanisms allows researchers to map out microbial evolution and deeply, deeply investigate how antibiotic resistance forms in the first place.
Now, what's really interesting about this slide is how cutting-edge research aims to fight back. To tackle the superbug crisis, scientists are actively looking for ways to block this gene transfer process directly in pathogens, literally stopping them from sharing resistance in the first place. They're developing CRISPR-based gene control systems designed to target and completely eliminate these specific resistance plasmids. By monitoring how these genes spread and designing safer microbial therapies, we're basically using the very mechanics of HGT to fight the problem it creates.
As we wrap up this explainer, I really want to emphasize this core takeaway straight from our source material. Horizontal gene transfer enables bacteria to rapidly acquire new genetic traits, making it a major driver of evolution and antibiotic resistance. It is so crucial to grasp this dual nature. HGT is the incredible engine of bacterial evolution that sustains life's adaptability, but yeah, it's simultaneously one of the greatest medical threats we face today.
Which leaves us with this final, provocative thought. As bacteria continuously trade their genetic secrets in this vast, invisible economy, will our next generation of CRISPR-based therapies actually be enough to outsmart the superbugs of tomorrow? It's an ongoing, invisible microbial battle happening right under our noses, and understanding it is the absolute first step to winning it. Thank you so much for joining me on this explainer. Stay curious and keep learning.